Dear students, today we are going to study the movement of water and solutes across the plasma membrane. First of all, we'll take up osmosis, that is movement of water molecules across a semi-permeable membrane. This is effected by the concentration difference of water across the plasma membrane. The water molecules moves from low osmotic pressure, that means high water content side, to the high osmotic pressure, that is low water content side. You can see in the slide that on the left side water is written and the movement direction of movement of water is depicted over here, and you can see that water is moving towards NaCl solution, that means the side which is having more osmotic pressure. What is this osmotic pressure? It is the pressure required to stop the osmosis, and the pressure difference across the plasma membrane is same as osmotic pressure. Why this happens? Because the cells, they are always laid in a fluid environment, and the water net movement of the water depends upon the concentration of the solute in ICF or ECF. If ECF has a more solute concentration, then water will move outside, and converse will happen in case of more solute if present in ICF. And so this osmosis depends upon the concentration of salt. Depending upon the concentration of the salt, the solution can be of three types. One is isotonic. What is this isotonic solution? When the osmotic pressure of extracellular fluid is equal to osmotic pressure of intracellular fluid, such a solution is called as isotonic solution. That means there is no net movement of water because solute concentration on both the sides, that is ECF and ICF, has a similar magnitude. Now the movement of water across the membrane starts in hypotonic solution, but in what direction? Let us see. When ECF is less concentrated, that means more water, then ICF, then water will start moving inside the cell and cell will get swelled due to end osmosis. So when the cell is laid in a hypertonic solution, less concentrated solution, then the movement of water will be from outside to in, and the cell will swell. This is called as endosmosis. Then we have this hypertonic solution. Let me move the slide. I don't know why it is not moving. Then come to this hypertonic solutions. When the ECF has higher concentration of salt than ICF, then, as I've told you in the beginning, the water will diffuse out of the cytoplasm, and this procedure is called as exosmosis. And exosmosis occurs in hypertonic solution, and endosmosis will occur in hypotonic solution. Then I should acquaint you with one terminology which is called as osmole. What is this osmole? To express the number of particles per unit volume is expressed in terms of osmole. We don't use grams. We use osmole. One osmole is equal to number of molecules in 1 gram molecular weight of undissolved solute. For instance, in case of glucose, 180 grams of glucose, molecular weight of 1 gram of glucose is equal to 1 osmole. When 180 gram of glucose dissolved in 1 liter of water, its osmolarity is 1 milliosmol per kg. Okay, so this was the osmosis. Osmosis is related to the movement of water, and the movement of water, net movement, is caused by the difference of the solute dissolved on either side of the membrane. Then another term is diffusion. Movement of ions when happens across plasma membrane from higher to lower concentration without the use of energy, that is energy independent event. Such a event is called as diffusion. Now we have various kinds of diffusion. The first one is simple diffusion. In the simple diffusion, we have transport of metabolites via pores without the use of carrier molecules is simple diffusion. In simple diffusion also, as I've told you, there is no requirement of ATP, or it is also an energy independent procedure, and the movement of the metabolites is from higher concentration to the lower concentration. How does this movement cause? It is caused via static pores, which are around 70 to 20 angstrom, and they are pores present between the integral proteins and the small molecules. They can pass across through such static pores, and they are permanent in nature. However, on the other hand, there are dynamic pores which keep on appearing and disappearing, and this is caused due to translational movement of phospholipids. I have told you in the beginning when I taught you the fluid mosaic model. It is due to the random movement of phospholipids. Now, on what factors does the rate of diffusion depends? The rate of diffusion depends on the size of molecules and also the concentration gradient across the plasma membrane. If the size is too large, the rate of diffusion will be brought down. On the other hand, when the concentration gradient is too high, the rate of diffusion will be raised up, and if the concentration gradient is brought down, then the rate of diffusion is also brought down. An equation is given. What is the permeability? Permeability is equal to K into D upon T. This K is partition coefficient and D is the diffusion coefficient and T is the thickness of the membrane. So diffusion, one kind we had studied, that is simple diffusion. Now we have another kind of diffusion which is called as facilitated diffusion, and this diffusion also does not depend upon the energy. That means it does not require energy. However, it requires a specific carrier protein which is called as permease, and it acts as passive conveyor belt. And what is this facilitated diffusion? Is a special type of passive transport in which ions or molecules crosses rapidly. It is more rapid than simple diffusion because of specific carrier protein which transfers it, but the rate never increases the rate of change of conformation of permeates. Whenever whenever we increase the solute, the facilitated diffusion will rise up, but not above the level of change of conformation in permease. Now the characteristics of this facilitated diffusion. The rate of transport is greater than simple diffusion, as I have told you right now. Rate of transport increases with increase in concentration gradient, but never exceeds the rate of change of conformation of permease. See the kinetics. You can see the graph that the rate of diffusion or transport in facilitated diffusion, it rises up with increase in concentration of the substance. It rises to an extent which is called as Vmax. But after Vmax, the rate of diffusion does not rise, because the rate, it is limited by the rate of change of conformation of permease. So this is the equation number one. S is solute outside, permease is present, and it gives solute permease complex, and S is transported inside. So it is depicted is as V is equal to V max upon 1 plus constant upon Km. Km is the binding constant for the substance to permease. Vmax is the maximum rate of transport of the substance, and C is the concentration of solute outside the cell, and V is the rate of transport. So these are the two kinds of transport. Osmosis was related to transport of water, and that depend upon the concentration of the solute on either side of the membrane, whereas diffusion depends upon the concentration of solute itself. It is energy independent procedure. The simple diffusion does not require anything else, whereas facilitated diffusion is carried out with the help of carrier proteins, which we also called as permease, because it permits the solute inside. These are special kinds of proteins which acts as passive conveyor belt. Why passive? Because no energy is required. And permeases are specific. They are specific to each kinds or species of ions or molecules. So dear students, this is all in this lecture. In the upcoming lecture, we will take up active transport, which is also very important. And thank you so much.